And today we will see what are the different types of database. For example, this portion we have covered in the last class. What is a generalized database and what is a specialized database. And now we have to see what is specialized database. Till hardlink connection, I think I have explained. So going to the DNA molecular database, we have basically two different types of databases. One is a primary database, another is a secondary database. The difference between primary and primary database will be containing the original sequence information. For example, you take a species and we send it for DNA sequencing, and they will give you a sequence. That sequence is known by the name primary database. So original sequence information is known as the primary database. Whereas there is another type of database that gives you an additional information in addition to the original result. For example, in the case of this second type of database, which we popularly called by the name secondary database, there is some explanation about that sequence. There will be some additional information about that sequence. Then we call it by the name secondary database. So these are the basic difference between primary database and secondary database. So in this secondary database, it will be containing annotated data. Annotation in English, you have studied about annotation. Annotation means an explanation of a word or a paragraph. They will give you a line and they will ask you the annotation. Earlier, that kind of questions are there. So annotation means giving an explanation. Explain something. So in the case of annotated data, in addition to original sequence, that they will also give you some explanation, some additional information. Then that type of database are called secondary databases. So these are the two basic type of databases that we can see. Then we have to see about few other information like LANL. LANL means it is an important laboratory in America. Los Alamos National Laboratory. And similarly, there is another famous laboratory in the European countries or Germany, that is known as EMBL, European Molecular Biology Laboratory. And it is located in Heidelberg in Germany. So these are the famous laboratories working in the area of bioinformatics. One is LANL and other is EMBL, and these are keeping the databases of various type of sequences. Then we have different type of, under the primary database we have NCBI, then we have SysProt, then we have PDB. These are some famous type of databases, and we will see these databases one by one. And under the secondary database, there is OMIM. OMIM full form is Online Mendelian Inheritance in Man. So it is a database that gives you some information about different Mendelian diseases affecting a human being. Then we have BDB genome database, SNP single nucleotide polymorphism. So for the time being, you understand what is a primary database and what is a secondary database. In the case of primary database, it will be containing only the original sequence information, whereas in the case of secondary database, in addition to original sequence information, it will also give you some information about the some additional information about that sequence. Then we call it as secondary database. Then in the next paragraph, next slide, these are the very famous or most popular type of databases. I will show you what is NCBI, what is DDBJ, then I will explain that. Now, we are going to the NCBI. Now, if you type NCBI in Google, you can see that this is the National Center for Biotechnology Information, that is NCBI. So this database is known as the This is a very famous database. So here we will get a lot of information about different types of sequence. This is the most important type of database. Now, if you take this database and go to the nucleotide, here you can find there is an area called nucleotide. And here if you put insulin, then immediately it will bring a lot of information about the different type of insulin. For example, this is a sequence database, the sequence of insulin. This is Octodon degusus, Aplysia, it is a slave. This is another animal. Then camel dromedaris, and their insulin. That's how the insulin of that. For example, if you click this one, it will immediately show you the sequence of camel. How the sequence of camel. See, this is the sequence. You can see that. So this kind of information will be available from NCBI, and you are also going to do this as a practical. In your practical, you have to find out how to search for a database. So here I am searching for the insulin gene of a camel, and you can find these are the sequences present in the insulin gene of camel. So it's a very long sequence. Now we will take another very small sequence. For example, this is one in Aplysia, and if you click the FASTA sequence, it immediately show you it's a very small sequence. So this kind of information is stored in the database known by the name NCBI. So going to NCBI, its full form is National Center for Biotechnology Information. It's a very famous laboratory. And this National Center for Biotechnology Information is actually a part of National Library of Medicine. National Library of Medicine. That is also another famous institution. That NCBI is situated in Maryland. Maryland is a place in USA. And they have established that site in 1988. In 1988 they have established this NCBI. Now, what is the importance of NCBI? Now, in the case of NCBI, they house a genome sequencing data called GenBank. GenBank is the most important facility offered by NCBI, and we will see how the GenBank works. I will show you that. In the case of NCBI, now, for example, they have a search engine. That search engine is known by the name. For example, we come to the NCBI original page of the NCBI, and this is they have a search engine. Just like if you go to Google, you have a search box, and that is Google search engine. Now here they have a search engine. That search engine is known by the name GenBank. Now GenBank or they have a database that is known by the name GenBank. For searching that GenBank database, we have a search engine called NRES search engine. That is the NRES search engine. Now if we type the name of any animal, for example, we want to get the some information about sequence of information of paramecium. Paramecium is a very small animal. Immediately it will bring the information about the paramecium. So these are some information which are available with the NCBI. They see this is a paramecium. So they also have a taxonomy browser, and if you click that, you will get a photo of that. We can find all the 17 genomes of that. What are the species of paramecium here? So a lot of information they are providing with you, and this is known as the GenBank. GenBank will be containing a lot of information. Now if you click that, this is all databases. But if I am taking only nucleotide, for example, I want only about the nucleotide details, and if I again type a paramecium, that will be only bringing out the information of nucleotides. See, so this is how the this is a very small paramecium, and I go to the FASTA sequence. I will tell you what is a FASTA later. So here we can find a small sequence. So this is the search engine that NCBI is using. GenBank, just like we have a Google search engine, they also use a search engine known by the name Entrust search engine. So that is the Entrust search engine. That is the importance of NCBI. And we will see other points regarding NCBI. In the use of NCBI, and they have some kind of GenBank coordinates with individual laboratories and sequence database such as EMBL and also DDBJ. EMBL, I told you, it's a laboratory situated in Germany. DDBJ is situated in Japan. So any information produced or accepted in the GenBank database will be immediately coming to Germany and also to the Japan. So there will be simultaneous updating of information. So same information will be updated at the same time in all three laboratories. So if you check that their website, their database, you will be getting all the information that we have uploaded in one database. So this is regarding the NCBI. Now here you can find some other information. 1992 they have NCBI grown to provide other type kind of database. So the original and the most important database of NCBI is GenBank. But not only GenBank, NCBI now contains so many other types of databases. For example, here you can find online Mendelian inheritance in man. If you type that online Mendelian inheritance in man, you will get a database. I will show you how the online Mendelian inheritance in man. It's a database. So this is the database. It will be showing the details about the Mendelian diseases affecting human beings. Now I will show you that how this database will be appearing. Then let it come. Then it will also give you some information, molecular modeling database. That is another database. Then that will give you taxonomy browser. Earlier I show you how the taxonomy browser, there as in the use of paramecium. Then NCBI has a similarity search engine. That is known by the name BLAST. I will also show you how the BLAST. See, this is the OMIM. If you want to donate, so this is the online Mendelian inheritance in man. Now we are going to type one, sickle cell anemia. Let's see what are the information that it will be bringing on. So sickle cell anemia is a Mendelian disease. So here you will get so lot of information about the sickle cell anemia. This kind of information are available. So we will be getting lot of information. What are the type of work, research works published in that area. So these are the information that will be available from the online Mendelian inheritance in man. So that is another important functionality or function of the NCBI. So basically NCBI is working to provide GenBank. GenBank is the most important and most useful and most powerful database. Because it is just Google. You type your name, sometimes if you put anything in your Google, immediately it will bring. If you type my name, it will give you a lot of information about my publication. So once you publish something in internet, that data will be available in internet and immediately it will search all those information and will bring it to you. So just like that, NCBI is a very powerful database. Now another function is the BLAST. Now I will show you what is a BLAST. BLAST is like now taking the similarity. Now in case of Google, they have a function called image search. So if you take the flower, if you take a picture of a flower and go to image search and paste that flower there and when we upload that flower, it will immediately show similar flowers. So that we can easily identify what is the flower that what is the type of species of flowers that we have taken a photo. So that is an easy way. The same way we can also check in the NCBI. I will show you how the BLAST is working. Now just go to the NCBI BLAST. Here we have an information. Just imagine that this information, we only have this sequence with us. This is a sequence. Just imagine that now we know it is a sequence of paramecium. But if you don't know that this sequence belongs to paramecium, how you are going to find it out? That is what I am going to do now. Just go to the NCBI. Now Google. Now here I am going to type NCBI BLAST. So this is a BLAST. What is the full form of BLAST? Basic Local Alignment Search Tool. That is BLAST. Now in the use of BLAST, what I am going to do is that I am going to make a search. I want to find out this is the sequence that is with me now. I want to find out to which animal this sequence belongs. That is my aim. Now what I am going to do is that here we have this we have a NC BLAST search engine. In that we are going to copy that information and see what will happen. Okay, so we have different types of BLAST here. We can go to the TN BLAST. So here we have different types of BLAST: nucleotide BLAST, protein BLAST, BLAST TX, T BLAST. And for our convenience, we take the nucleotide BLAST. That is the most simple type of BLAST. Now in the nucleotide BLAST, I am going to paste that sequence information, and you can see what will happen. So I am going to copy this information here. I right click it and copied it. Now when this page is loading, there is a search box. In the search box, I am going to paste that information. Okay, now I have pasted that information. Now we will see what will happen. The only thing that I am going to do is I am going to click BLAST. So when I click BLAST, we will get the result. It will going to search just like we put your name. You put a keyword in Google search engine. For example, if you put Kuravalangar, it will be bringing you all the information about the Kuravalangar. Just like that, it will be bringing all the information and about this sequence or similar sequence. Now we will see what will happen. Now it is now checking. So let's see how much time it will be taking. It depends on the speed of the internet. It will come very fast. Otherwise it may take some time. Now the result is available. Now see, they are bringing the result. So what is the sequence that here? Paramecium tetra aurelia sequence. That is the sequence, and the number is and there is a GenBank number AL51255.1. That is the code number. Now let's see what is the result. See here they have bought this information. Now if you click that, it is showing 99% coverage, 95% similarity, then 97% identity. So if you click that information, we can see that it is the Paramecium tetra aurelia hypothetical protein, the same thing that we have got here. Then plasmid T015021. Let's see if there is some difference is there, but still there is 97% similarity. See, there is 97% similarity. So this way I will show you one more. For example, I am going to search about insulin. Now this is the small insulin, octadone. Now I will be copying this information. I am now copying this information and going to paste that information again in the search box. So let's see what we will get. So now I am going to paste this information here, and again I am going to search BLAST. So it is going to it is searching, and it will be bringing out those information that you are looking for. The 9 seconds. So this is a practical that you have to do. You have to go to NCBI and find out the sequence of a nucleotide. Then you have to also go to BLAST and find out how to check for a sequence, how to find an unknown sequence. That is what I am showing you now. Now it will be bringing the information. Let's wait for some time. See, they have, so all the matches are here, and on the top you can see that octagon degausses insulin mRNA complete CDS. Let's see, octagon degausses insulin mRNA complete CDS. So this is the information that we have supplied, and it is bringing to us the information that this unknown sequence belongs to the octagon degausses insulin. So that way we can find out an unknown sequence. For example, you get a meat, a small piece of meat. We don't know to which animal that meat belongs. What we can do, we can go take that small portion of meat to a biological laboratory or a DNA laboratory. In that DNA laboratory, they will take a small cell or small portion of tissue, and from that tissue they will be taking the DNA. From the DNA, they will give you a sequence. If you take that sequence and put it in the NCBA BLAST, it will be searching all the sequence in the database and find what is the most probable sequence. So by that method, we will be able to find out an unknown animal. If you don't know, the name of an animal or name of a species, you can easily find out if to which species or which phylum or which family this animal belongs. So that will be making a DNA similarity search. So that is a function of NCBI BLAST, and NCBI BLAST is a very important program, important function of NCBI. They have, one is GenBank, and second most important function is NCBI BLAST as a search engine. Then here this is the picture of the NCBI. Now another type of is the DNA Data Bank of Japan. That is the second data bank, that is DNA Data Bank of Japan. Now let's see what is DNA Data Bank of Japan. I will show you DNA Data Bank of Japan. Now if you go to the DNA Data Bank of Japan, here you can find DDBJ. That is the DNA Data Bank of Japan. Let's see what will happen. See, this is DDBJ. So this is a site that they are giving you. So we are going to get information about DNA data. What are the information about DNA database, Omega? This is located at National Institute of Genetics in Japan. It is a very famous institute, just like we have Indian Institute of Science or AIMS, All India Institute of Medical Science. They also have a very famous institute, National Institute of Genetics, that is located in Japan, and they are also keeping a database which they have an international collaboration under INSDC. So they will be transferring information or sharing this information with GenBank and also European Molecular Biology Laboratory. Then about their activities, they have started in 1986 at NIG, National Institute of Genetics, and that is the only sequence database in Asia. So in India we don't have a sequence database because Indians are late starters in this bioinformatics, because bioinformatics started in the USA around 1970 or 80. But in India, that bioinformatics were started functioning after 2000 only. Then other information about DDBJ is that they are supported by their Ministry of Education. Then I will show you what is the nature of this is the nature of the DDBJ. Now if you want to submit some information, for example, we will submit some one information, then we have to follow certain procedure, and we have to search analysis. If you want to search something, you can go to this way. I think this is the technique for searching. And since I am not very familiar with DDBJ, I don't know how to which is the page, how to submit. See, we can say that page is not available now. But DDBJ is actually a database operated by National Institute of Genetics in Japan. Then another famous database is their type of database is this is the earlier view, OMIM. Regarding OMIM, I have already shown you. OMIM is a very comprehensive database, and that will give you a lot of information about Mendelian disorders. Mendelian disorders are basically genetic disorders. Different types of genetic disorders are the disorders which are based on the dominant successive trait are known as Mendelian disorders. And they will be showing a relationship between phenotype. For example, if a person has a particular gene, then they will have morphologically that person will be having a certain defect. Sometimes cleft lip. Sometimes some people are at the time itself they will be albinos. They won't have any melanin pigment. These are all what we call Mendelian disorders. So this database will be giving you information about Mendelian disorders, and it was started by one person known as Victor A. McCusick, and he wrote first produced a book called a Catalog of Mendelian Traits and Disorders, which was later, it was a 12-volume book. That book was later converted into an online catalog that is known by the name Online Mendelian Inheritance in Man. So this is the online version was started in 1985. In 1985 we still don't have a internet even. So they are they started all this at a very early stage. That is why they are still far advanced. They are far ahead of us in this kind of technology. Then by collaboration between two National Library of Medicine, then medical library at John Hopkins is also a very famous medical college or medical institution in the world. So these are the very famous. So it's actually a collaborative work of two major institutions like John Hopkins University and then National Library of Medicine. So main purpose of OMIM is that OMIM will be giving you information about Mendelian disorders. So today we will be stopping with here.